Thanks everyone for making it this far, and welcome. I work in the life sciences, and this is an area where we've increasingly adopted R. It's very much on the rise where I work. And not only that, we do things with R that are increasingly computationally demanding. And today I want to share with you some of the hardest kinds of things that my colleagues and I do, and how we manage that work with pipelines. Before I begin though, you may want to queue up, oh, thank you. My computer slept. Before I begin though, if you'd like to ask a question at the end of the talk, and if you'd like more time to queue it up first, then just head to this link right here. I'll show this at the end as well. So, my colleagues and I do a lot of modeling and simulation, and every time I have a project like this, I think about one of my favorite webcomics. So, we're in an office, the hiring manager said, the programming manager says, get back to work, and a couple of developers are out here messing around, saying, no, my code's compiling. And the manager says, oh, okay, carry on. And it's one of the most relatable parts about professional software engineering, where you write a program to do something, but before you actually can use it, you have to let it compile, if it's C, C++, or Fortran code. And while it's compiling, as a developer, there's nothing for you to do. You just have to sit there and wait for the computer to do its thing until it's done. And now, most of us are not professional software engineers in R. A lot of us are statisticians. Some of us are mathematicians, ecologists, economists, geneticists, and all kinds of quantitative scientists. But we can relate to this. We can very much relate to this still, because instead of my code's compiling, it's my model's training, or my simulation is running, and there's nothing I can do until it's done. And instead of horsing around having sword fights in the office, we experience pain when this happens. So it is painful to get ripped away from our regular work, to wait for this thing where we were in a groove and we were in a flow before, now take an eternity to actually complete so we can move on to the next step or the next iteration, wherever that is. And it's extra painful because these kinds of projects are almost never one and done. There are all sorts of reasons to go back and update the project until we get it right and until it's complete. And if we have to repeat this waiting time every single time, it can feel like we're King Sisyphus in the underworld pushing up that boulder up the hill constantly. And it's hard to reach that point where we have results that we're confident in that we can share. And so one of the places that my colleagues and I experience this the most is simulating clinical trials. It's something that we do a lot. It's a kind of work that's critically important in our process. It plays a really important in the whole scheme of things in drug development. And it's this enormous pipeline that takes decades, where we begin with thousands of molecules in the discovery phase, and through in vitro experiments, in silico experiments, biological assays, we narrow it down through preclinical studies and clinical studies, down to a small number of treatments that are safe and effective for humans to use on a routine basis in a clinical setting that doctors can prescribe. And we have this deliberate bottleneck at the clinical trials phase to make sure treatments are safe and effective. And this clinical trials stage itself has, as we're funneling down the number of prospective treatments, we are expanding the resources that we're using in the trial, and we're asking for more and more participation from people who enter these trials. And we have some critical priorities to balance. We are trying to create treatments for the future, but we have the utmost responsibility to patients in the here and now who are actually in the trials. Their safety and well-being come at the highest importance, which is why it's just so important to fine-tune and design trials as intelligently and parsimoniously as possible. And one of the goals of clinical trial design is to make sure it can make the correct decision when the time comes. If the treatment works, we want to show conclusively that the treatment works. If it doesn't work, we want to show that it doesn't work. And this involves a very careful process, quantitative and empirical, where we compare a bunch of different alternative designs and we simulate them and we compare their ability to make the correct decision. And how we do this is we simulate virtual patients. Those can be just a numeric vector of outcomes, for example. Not always numeric, but things that we see a lot are, for example, ADAS-Cog13 scores for disease progression in neurology, or PASI scores in the case of psoriasis. These are outcomes that follow a distribution that, if in a disease state that's studied a lot, we can come up with reasonably good data models for. And for each set of virtual patients that represent a hypothetical trial, we analyze them with the models that we plan to run when it comes time to execute the trial. And then we repeat this thousands and thousands of times to get operating characteristics or metrics that are aggregates across the entire simulation. We do, of course, look at individual representative simulations too, but in aggregate, that's where we look at power and type 1 error, et cetera. And this is, of course, very challenging. The kind of simulations I'm talking about today are, of course, done with R. And these are going to be the most complicated ones. We already have internal Shiny apps and commercial off-the-shelf black box programs for the simple cases. So the ones that we even bother to code up with in a custom way are already going to be the most challenging, whether it's an advanced operational feature like response adaptive randomization or a complex Bayesian model. We're going to code those in R. That's where we take on the biggest challenges. And of course, these have long execution times, not only because of the thousands of simulations we're running, but the caliber of the models that we choose to run in these simulations sometimes. And of course, frequent changes, updates to the project, new questions come in, new priorities come in all the time, and we have to be able to adapt. So let's go through an example trial. Everything about this example is hypothetical because I'm borrowing public data for this in one stage of the simulation exercise that I'll talk about. But it is representative of the kinds of problems we face and the kind of approach that we take to tackling them. So suppose we're studying an extremely rare autoimmune liver disease at a severe stage of the disease where a patient is likely within the next few years to need a liver transplant. And this is a phase 2A study. This is typically one of the first studies we do after the phase 1. So we're first looking at efficacy. It's a 2-arm double-blind study of this treatment versus the standard of care. And we randomize the patients 1-to-1. This is a rare disease. It's a phase 1. It's a very small study. This is exactly the sort of situation where it'll really help us to use a powerful Bayesian model, and in this case, a Bayesian joint model. So the model in this example has been argued in the literature that it is more more power to detect treatment effects than a typical survival model. This joint model is a model in 2 parts. We have a regression on a longitudinal biomarker, and this expectation of the expected outcome from this model is actually part of the survival function in this. This part of the hazard function in this survival model. That's what makes it a joint model. There is an association between those 2 parts. And of course, we have our log hazard ratio theta, which quantifies the treatment effect in this case. And because this is a Bayesian model, we have the advantage of straightforwardly quantifying things that we wish to, that'll help us plan future stages in the clinical program. So we can straightforwardly ask, what's the posterior probability that our hazard ratio is small enough? And our threshold is that we want the infinitesimal risk of needing liver transplant or experiencing death to be 25% less in the treatment group than in the control. And so we're declaring efficacy in our primary analysis if we're at least 80% sure that that probability is that hazard ratio is low enough, and we declare futility if our belief is very low. And let's get to the operational characteristics of the trial, because this has to do with the questions that this simulation exercise is trying to answer. So this trial is going to take a really long time to accrue, common in rare diseases. And so we want to choose an adaptive design. Somewhere along the execution of the trial, there may actually be enough evidence to make a decision early, before the original end date. And this could save extra patients from being enrolled. For example, if we have enough information at some point to know that the drug doesn't work, then it would be a good idea to stop the trial for futility and save patients from being exposed to a drug that at that point we know doesn't work. And so that's the goal of this simulation, to try to find out when this futility interim should be. If it's too early, then it's just going to be inconclusive. If it's too late, then we may have exposed more patients than we need to. And so that's what we're trying to simulate. We're trying to find a suitable number of events to run the trial until to balance those things. And the way we're going to do this simulation is called a probability of study futility calculation, and it's a Bayesian approach to this kind of simulation. We are going to simulate 10,000 data sets for each event threshold, and we're not going to choose one fixed assumption about what the, quote, underlying true hazard ratio is. Because what does futility really look like? It could look like a hazard ratio of 1. Treatment and control are basically respond the same. But it could be a hazard ratio of 0.9 or a hazard ratio of 0.8. It could be statistically significant, but not practically significant enough. And if we fix a hazard ratio of 1, or really any fixed hazard ratio, we could go in overconfident in a number of events that's too small. So we want to have some realistic level of uncertainty around our assumptions about what the underlying hazard ratio is. And so what we're going to do is we're going to simulate hazard ratios from what's called a design prior, which in this case is the posterior, what I'm taking as the posterior distribution of that hazard ratio from the same model on a real data set. And I'm using, for this example, some public data from the survival package on PBC, primary biliary cholangitis. And this is from a Mayo Clinic trial from decades ago, in this case, where the drug didn't improve survival. And what we get out of this is that design prior. We get a distribution for the hazard ratio whose center is around 1. So, time to implement this in R code. Before we get to the pipeline part of the talk, let's set ourselves up for success for such a large, intimidating project. And I really love functions for this and recommend functions all the time. One of my favorite quotes from John Chambers, the creator of S, everything that exists is an object, everything that happens is a function. And a lot of us in here have already written our custom functions and write them all the time, especially package developers. But I think it's a leap from writing functions for some things, like some things that happen are functions, which is the case if you're coding something in the tidyverse and write functions one-off functions to use with dplyr or per. This is another level of that where we're treating everything as a function. And it's natural because in a simulation or in a data analysis, the things that happen truly are transitions. They're transformations from data to fitted models, transformations from fitted models to summaries. In a simulation, it's the same thing. Everything that happens is a function. From transforming public data to a posterior distribution on the hazard ratio, which is our design prior, to simulating one replication of the trial, to visualizing the results altogether. And these functions have clean inputs and nice, tidy, rectangular outputs. So you input a hazard ratio that's going to come from that design prior, and it could be something very low, in which case we'd have a high efficacy posterior probability, or it could be something really high, maybe around 1, that puts our efficacy probability really low. And if we were to put this in an imperative script altogether, which is a natural thing to do, it's a natural thing to think about what does a run everything script look like in this situation. Well, we fit we call the draw hazard ratios function to fit that model to the public data. We get a vector of hazard ratio draws. These are Monte Carlo samples from the hazard ratio. For each event threshold and each hazard ratio that we drew, we run one replication of the trial. That's this tidyverse pipe chain. And then we visualize the results altogether, that entire data frame of results. This may look simple because I simplified it for this talk, but in practice, it's way more complicated than this. And there are many, many challenges of putting it all together in a workflow. And those challenges are exacerbated by the caliber of the computation that happens inside of an individual trial simulation. So a lot of that comes from the Bayesian model that we're using. So we're fitting this advanced joint model that comes from the rstan-arm package. There are a few R packages that implement joint models, and this is one of my favorites, the stanjm function and rstan-arm. And that is implemented with Stan. How many of us use Stan here? Oh, wonderful. For everyone else, Stan is a beloved probabilistic programming language, popular among Bayesians. And it runs Hamiltonian Monte Carlo, which under the hood is a physics simulation. So how it intuitively works is you take the log, you take the density of the joint density of the data and parameters of a model together. You take the minus log of that, and in a well-behaved case, you get a nice bull. And the physics simulation boils intuitively down to putting a marble on that bull, flicking it around, and then taking iterative snapshots of the trajectory of that marble in time. And those samples of those locations give you the same properties as a collection of samples from the distribution you're after. Bayesians are very concerned with sampling from the posterior distribution. That's computationally one of the hardest things that we do. And it's an inherently difficult problem. I know this is like a two-dimensional parameter space that I'm showing, but in reality, it's like hundreds of dimensions, and for a lot of models. And Stan does an incredibly good job of being efficient. rstan-arm as well. This runtime of just under 3 minutes to fit on the public data actually isn't so bad. And each simulation is even quicker, about half a minute. But if you put that all together, you might not, if you were to run this on your local laptop, you would be done in 2 weeks. And that's just one round of this. So you might have to deal with, not only the responsibility of a huge computation, but the responsibility of repeating it over and over again. And to put all this into a workflow, that's an additional challenge. We've often heard recommended that you can divide a project-oriented workflow into different scripts or different reports. This is really nice for a simple case. But I've really, in the past, my colleagues and I have felt the pain of, for this size of project, it breaks down for us very quickly because there's a lot of engineering to do on top of it and a lot of change to manage. it's hard to engineer, just the data storage alone. To manage that is very complicated because the reports have to share data amongst themselves. It's up to us to figure out a way to do that. There's also the change management. We'll have extra questions, extra steps in a pipeline. It's really easy to start simple and then have to awkwardly make it more complicated later. And then for the case of the simulation, when do we rerun this? And how can we avoid rerunning steps that we don't need to rerun? It's very painful to just run everything from scratch. We would rather skip work that we don't need to run, but it's very difficult for a human to make that kind of decision. And so pipelines are a great way to navigate that. Now, so far in this talk, I've used pipeline, the word pipeline at least once informally. I think that's how we usually use the word pipeline. We even talk about pipe chains in the tidyverse sense as pipelines. For this talk, what I mean by a pipeline is a formal group of tasks with some restrictions. And it may seem overly pedantic, but for this kind of project, we get a lot more out of it than we put into it. These restrictions really give us a lot of power and save us from mistakes. So in a pipeline, as I'm defining it here, and for the purposes of targets, each task has dependencies that it draws from, a command that it runs, and some output that it produces. And there's restrictions on how tasks relate to each other. So a task can't modify other outputs other than its own. A task can only modify its own output. So the plot task cannot go back and modify the task of the trials. And a task can't eventually depend on itself. And what this means is if you put all these rules together, then you'll end up with a nice mental model of your workflow as a directed acyclic graph with no back edges that you can easily draw out with nodes and edges like this. And pipeline tools, they give you a lot of power. They most have some way of skipping work that you don't need to rerun, whether it's resuming a workflow or doing something like make, which really aggressively is about skipping tasks. And there's a lot of power in parallel computing, distributed computing as well, and it gives you evidence of reproducibility. And there's a huge ecosystem of this, especially outside R. Many of us in this room use make. Many of us have heard of Airflow. Those are really prominent tools. And in R, we have targets. There's also a new pipeline tool coming out called Rexpress. But targets has been around for a bit longer, and it's a bit more of a generalist. And targets makes a huge effort to integrate with R and to integrate with the conventions and tools that are in R and how we expect to work and how we work best. So it retains even if it spawns parallel processes, it still retains the comfort of your local R session. It focuses on functions and the relationships between functions rather than scripts. It's a more natural, if you're already working in a function-oriented style, this is very natural. It integrates with Quarto, it has literate programming features, and it still gives you the power of distributed computing. And so let's look at implementing this trial simulation pipeline that I described before. So the structure may look a little bit like a package or a research compendium. You have scripts that write the custom functions that express this trial simulation. And at the top level, you have this _targets.r file, which is kind of like a configuration file or a make file. How many of us use make in this room? So for those of you who are familiar, this targets file is like a make file or a YAML configuration file, except that it's written in R. It's written in R for not to be able to, not because targets expects you to run this manually, but because if you're already working in R, programming even the configuration in file in R is very natural. That's not only where you define the tasks of the pipeline, it's where you set your options and you set up the environment where it's going to run. And a bit about setting up that environment. One of the great things about pipeline tools in general, not just targets, is that there's the graph, that mental model, gives you a lot of power for distributed computing. And that is completely separated from the individual tasks that you'll code up. When you code up those individual tasks, you almost never need to code in the logic and the tools for parallel computing at that level. Which really cleans up your code and helps you focus on the content of the statistics instead of parallel computing. So these are options that get set up front once. And this is a really demanding simulation, so I'm using 500 workers on a SunGrid engine cluster. And as far as the inputs go, these individual tasks, you'll give it a name and a command to run. And in this case, the command is just to list the event thresholds that we're trying. And this is really equivalent, conceptually equivalent to a function that just executes that command and saves it to disk. And likewise, with the hazard ratios, we're fitting the model to public data this time, except we take the function draw hazard ratios as a dependency, as input for this. And Targets has a feature called dynamic branching, which allows us to, in a function-oriented way, loop over the hazard ratios that we drew and the event thresholds that we're trying. And that's equivalent to, for each combination of hazard ratio and event threshold, do this over and over again. And the trials target is actually many, many targets on disk. There is no one file that stores all of the trials. But you can, if you refer to it symbolically and don't map over it in dynamic branching, then you will implicitly in targets combine all of them together. And under the hood, there's this magic that happens with the vectors package in order to accomplish that. It's very friendly with data frames. And that's the equivalent of visualizing all the results and saving the ggplot to disk. Lastly, in the pipeline, there's a step that runs a Quarto report. The target types package has a bunch of interfaces to connect targets with other tools and to write interfaces for doing common tasks in targets really easily. All you need to do is supply the source and of the report, and it'll render. And when the pipeline executes, it'll render it and then track the source file and the output file for changes. And this Quarto report. Targets tries to combine interactivity when you need it and automation for reproducibility's sake and for the sake of having confidence in your results. And so it nudges you to write literate programming reports that trust the pipeline upstream for the computation, and all that's left for the Quarto report to do is load results that you've already computed through functions like tar read, and to spend most of your time explaining it in prose. And that's sort of the balance that works really cleanly for a lot of pipelines we've seen over the years. And the graph, it's not only a mental model, it's a physical model that you can call up on demand because of how Targets analyzes your code base for dependencies. It's very into R. You don't declare dependencies manually. You declare dependencies implicitly by the way that you write your code and the symbols that you reference. So this plot command depends on the trials target that can be detected and is detected with the code tools package with static code analysis. And that's how Targets knows to draw a line that you see highlighted here from the trials to the plot. That's how it puts together the entire graph. As far as running the pipeline goes, there's this function tar make. So Targets is very much inspired by make. It has a lot of the same opinions. And so this function tar make runs the pipeline, traverses the graph, parallelizing when it can, and it may be spawning background processes, but fundamentally this is an R function. And if you're running in RStudio or Posit Workbench, then it'll run it as a background job and free up your console. And while this is happening, this is what's really going on in terms of the graph. And a lot of this is, the way the distributed computing works is orchestrated with the crew package underneath, powered by another amazing package called Mirai. And what crew tries to do is scale up workers in parts of the graph where there's a lot of work to do, and then scale back down when fewer workers are needed, because we're asking a lot of the cluster. It's a shared resource. We do want to be able to run hundreds of tasks at the same time, but we also want to scale back down as soon as we don't need those tasks so other people can use those resources. And so how this works automatically is in the targets.r configuration file, we set a maximum idle time of 2 minutes, and that'll help us scale up and down. And so what happens when we run this, when we're fitting the model to the public data for the design prior, we spin up a worker automatically in crew, because crew sees what is the demand of the tasks that need to be run and how many workers do I have running right now that are free to run new things, and it scales up accordingly. Same when we go to the simulations. So more workers are required, and so the workers automatically scale up accordingly. When we go to do the post-processing, the plotting and visualization, fewer workers are needed. We have one worker idling here in this case. And in the real case, there are 499 idling workers by this point. And so those automatically terminate after 2 minutes, keep system administrators happy for the cluster, and keeps other users happy who want to take advantage of these same resources. And what you get is an end-to-end complete pipeline. And again, this combination of automation and encapsulation, which is not how we typically use R, versus the interactivity that we're used to in R, targets does really try to strike a balance, because there are functions to inspect the progress of the pipeline while it's running, graphically as well. Crew has functions to monitor the workers that are running, so you can keep an eye on your resource consumption. All of this is available from the comfort of your local R session. And likewise with interacting with the data. So ordinarily you'd have to remember, where is this data located? Did I misplace it? Because it's easy to feel like you have to engineer a project from scratch in this sort of situation, but targets has a central data store. You just call tar read, you give it the name of the target you want to read, and in this case, which branches you want to read, because this is a dynamic branching target, and it gives you the simulations that you requested. This would be true even whether it's located on disk or located in S3 bucket. So it is possible to balance the interactivity and automation when it comes to the needs of pipelines and of users of pipelines in R. And which brings us to this plot, which is the culmination of the simulation. And for each event threshold that we tried, for the futility interim, we see the number of patients enrolled, and we see the probability of futility, the probability of detecting futility correctly. And we see that while we have very different numbers of patients enrolled by this time, there isn't all that much difference in the probability of detecting futility, which may make us prefer earlier futility rules among the ones that we tried. So that's an actionable step. Clearly more work to do, but it gives us a start. And of course, the Quarto report nicely rendered HTML, and that refreshes when we refresh the pipeline. It can also be run interactively in your main session. Now, of course, like I said, there's constantly more changes that need to be done, questions that come in, bugs to fix, etc. So you iterate a lot on this project, and it's really good that we have the graph here, because we can let the graph be our guide for how to wrap our heads around all this change and to make sure that what we've changed is implemented correctly. So we add a new target because maybe a colleague asks, well, how long do we need to wait for this futility interim in each case out of the options that we've tried? And so we add this new plot years target to summarize the results that way. Because we correctly spelled our arguments, we have this nice edge that connects the trials to this new years target. And in this graph, we label things that are already up to date versus the things that need to be computed for the first time or recomputed. And when we run the pipeline with tar make, there's hours and hours of work that's skipped. 40,002 targets are skipped, and only that final years target is computed, saving us hours and hours of time. And we see a similar story here, where there's a huge difference in how long the trial needs to run up to this milestone for the different thresholds that we tried. Likewise, we go back and change things all the time. We fix bugs, we try new things, and where this happens most often is in the post-processing, the visualization. And with confidence, we can look back and see, well, what's changed, what is going to run next time. And we've spent a lot of time just changing the visualizations, changing the summaries, and then with confidence going forth and being assured that everything else is up to date, we didn't actually break our simulations as we're working on our plots. And so it gives us more trust in the results. And if we change an event threshold, for example, this appears to invalidate everything downstream, which in the general case might be. But if we were to rerun this simulation, if we were to call tar make again, it would actually only run 10,000 simulations, only the one corresponding to the event threshold that changed. Because of the caching and dynamic branching and everything, it'll skip those first 3 event thresholds, skipping about 75% of the pipeline. Likewise, it's aware of your functions and how your functions connect to one another. And I think R has been missing this for a long time because it's very traditional for pipeline tools to track individual scripts, but not individual functions, even though we're thinking about the code in those scripts rather than the containers around that code. And for example, you can tell that if we change how we're simulating the survival data, that affects everything downstream, all the functions that call that little function that we wrote, and so that invalidates all our trials. And in this case, we will need to run everything again. And at the end of the day, we have an up-to-date pipeline. And if everything's up to date, that's tangible evidence of reproducibility, literally at face value reproducibility, that these results can be recreated, evidence that these results can be recreated from the inputs, the code and data that we have in front of us now. Literal programming, containerization, also incredibly important parts about reproducibility. But this belongs in the reproducibility camp as well. So conclusions for this particular study, an interim at 40 events, it looks promising for this case. We clearly want to do more simulation to investigate that in a more fine-tuned way. We may even be interested in a group sequential design because of how long this trial is projected to run. But it gives us a really solid foundation to grow this project however we need to. And so pipeline tools, I think, have not only a space in the DevOps and software engineering camps, they really have a role in classical statistics as well. And I think we've been missing that for a long time. They save us, when working with a pipeline-based approach to classical statistical problems, they save us a ton of time, especially when we're iterating on this stuff. They provide evidence of reproducibility, and overall, you end up with a cleaner, more robust project, and it gives us all a lot of peace of mind and the ability to trust the results more. Tremendous thank you to the organizers of this wonderful conference. And I'd also like to thank R-OpenSci not only for adopting targets as part of its collection of reviewed packages, but also being a tremendous help for me getting started as an open source developer all those years ago. So with that, thank you for your attention.